Well, what we're actually looking at is we're trying to understand the biochemistry that occurs within cells, and in particular human cells involved in cancer. So when a signal hits a cell, you then have messengers or proteins that take that message from the outside of the cell to the inside of the cell, to the nucleus where the DNA is stored, and that tells the cell whether to switch on or off genes. But as of about 10 years ago, this was invisible, but we can now use fluorescence and luminescence, which are naturally occurring colours, to actually be able to visualise these events. And fluorescent organisms, for example, corals and jellyfish, or luminescent organisms, for example, some starfish and fireflies. And what we do is we actually use the genes, the fluorescent or luminescent genes from these organisms, attach them to our genes of interest in our human cancer cells, and therefore we're able to visualise these molecules of interest due to the fluorescent tag. So we start off doing molecular biology because we pick what gene of interest, so for us in our lab it's NF-kappa B, and then to make it fluorescent, so to attach it to that fluorescent gene takes a month, a couple of months, something like that. It's a long period of time of doing cloning, molecular biology, a lot of lab-based DNA techniques. Then once you've formed that fluorescent protein, you actually can insert that into your cells, or your cells of interest, which you've got maintained in your tissue culture lab. And then within a couple of days you can actually put those cells then on the microscope and actually visualise the movement of that fluorescent molecule, because the cells will take up that fluorescent molecule for a few days, and you can actually then visualise the movement of that in your cells using microscopy.
So what I'm doing at the lab here is looking at the role of the transcription factor called NF-kappa B in the cancer called neuroblastoma, which is a childhood cancer which has still got a very high mortality rate and chemo resistance. I've got the next 2 years here, and in that time I hope to find out how the inhibition of NF-kappa B pathway or the modulation thereof would affect the survival of neuroblastoma cells, especially in combination with chemotherapy agents.
I work in a group of about 12 people. We have students starting off that are doing PhDs. We have some undergraduate students as well, which come to us for 2-3 months. And then postdocs, so you could have a postdoctoral contract for 2, 3, 4 years. And then research fellows. And then we also have the professors and people involved in looking after the microscopy and technicians. So it's quite a varied group of all different levels. What we hope is, now that we can understand the patterns that occur within cells or within large populations of cells, we hope to be able to use chemotherapy drugs or cancer-inducing agents to try and understand how we can modify these responses that are seen in cancer cells. And eventually I'd like to take that further by looking at how cells interact, and then move on to tissues and understand how cells interact in tissues.
What excites me about my job is that every morning I might be getting up to do something new, which potentially is something that no one else in the world knows about, and which might one day make a difference to a child's life.
What we found is that what we thought was a simple switch, so the cell received a stimulus, these molecules moved into the nucleus and switched on or off genes, isn't so simple. In fact, the molecule of interest moves into the nucleus and then back out into the cytoplasm, and this cycles back into the nucleus and out again with a period of about 100 minutes. And this occurs for several hours. So what the cell's actually reading is the frequency or the period between these oscillations. So it's working like a cell language. It has to measure something for several minutes or hours to actually understand what to do. It's not a simple, quick event. And so what we want to do is see what chemotherapy drugs can do to affect these oscillations.